everyone. Thank you so much, Drew, to be with us today, and I'll let you take it away.
Thanks, John, and thanks, Carrie, too. Thanks to you both for organizing this and for hosting me today. I'm excited to talk about this topic. And thanks everyone who's attending online for your interest in my presentation and this topic and being able to join us today. So my name is Dr. Drew Harner. I'm an assistant professor of horticulture or viticulture specifically at Virginia Tech in Virginia. And today I'm going to be talking about some research my lab has been conducting looking at how the spotted lanternfly, which is an invasive plant hopper, can impact grapevine gas exchange and various photosynthetic traits. So to give a quick plug for my lab, I joined Virginia Tech in 2024. So I just wanted to give a quick background. I am a viticulturist, so that means I work with grapevines, specifically wine grapes mostly. And my lab really tries to focus on how we can improve the sustainability of wine grape production in Virginia and the Mid-Atlantic more broadly. So we do research focusing on various aspects of wine grape production, from viticultural practices to how different environmental conditions and stressors like the spotted lanternfly impact grapevine biology. And I'm also an extension specialist too. So I work with growers pretty closely and hold workshops from everything from pruning to different aspects of farm management and really just work closely with the industry here. So that's just a quick background and where I'm coming from, just to help frame my experiences and sort of this presentation. So the spotted lanternfly, maybe many of you have seen it, maybe you haven't. If you haven't, it's probably coming your way if you're in the US. And this is what they look like. This is the adult stage of the spotted lanternfly. It is a sap-feeding plant hopper. So specifically, it feeds on the phloem mostly and consumes phloem sap. And it's native to Southeast Asia originally, but arrived in the US in 2014 in Southeast Pennsylvania. And since then, it's been spreading throughout the Eastern US and now the Midwestern and Southeastern US as well. And this photo just sort of gives you a good example of what a bad infestation can look like. These are grapes that are ripening. This is sort of approaching harvest. And when you really get heavy infestations, they're all over the vine. And they feed on the vine through this little appendage right here. And they tap into the phloem and consume the phloem sap directly, and are pretty smart about mediating or modulating the plant defense responses to try to have this continuous consumption of phloem sap. So when they're feeding on the vines in quite high numbers, as you can imagine, they're removing quite a bit of phloem sap. So that's something to keep in mind as we work through the results and some of the things that I'm presenting. And since 2014, I mentioned it's expanded throughout the US. This is a map that Cornell, New York State IPM puts together of all the current documented infestations. So these are instances where people see them, report them, and then someone goes out and verifies, yes, there's a population there. And you can see at this point in time, almost in the entirety of Pennsylvania has populations. Most of Maryland or almost all Maryland, Delaware, New Jersey. It's expanding into New York as well in the Midwest. So 18 states altogether, including even down in the South or Southeast further south than us here in Virginia, in Georgia. And you can see Virginia right here. Where I am is west of Washington, right in this little corner. We've had spotted lanternfly since about 2018 or 2019, and it's steadily spreading into the rest of the state and where our major wine grape production is. And just to give you an example of what their life stage is like, if you haven't seen them before, they're one basically life cycle per year. Their eggs hatch in about May to June. And down here in Virginia, we sometimes get them in late April. And these what they look like in their first instar. They have four instar phases before they turn into adults, and they're really teeny tiny, like very small when they first hatch. And they typically hatch in vineyards in the very early spring. So vines will start to grow. We'll have maybe 3 to 5 inches of growth, and they're already hatching. So they're present in the vineyards through the entire season. And then they molt through these different life stages. And this life cycle was put together by New York State IPM. And these dates match more or less with what we see here in Virginia. It's a bit warmer here, as you can imagine, compared to even Pennsylvania and New York and Ohio. So we do get some earlier phenological development for them. But really, what I'm concerned about as a viticulturist and a grapevine biologist is the adults, mostly the adults. We're concerned about all aspects of the insect, but the adults are really what have emerged as the life stage that can really impact grapevine biology as well as other host plant biology, because they can feed on over 60 species of plants documented, at least native plants here in the US. So they're extremely cosmopolitan in their diets. But within that wide host range, there's really probably 5 to 10 species of plants that they feed on the most. And unfortunately, grapevines are within, that core group of host plants. The other ones include tree of heaven, which is also an invasive tree in the US, originally native to, I think, Eastern Asia. That's one of their primary host plants. And that's because they feed on that prolifically, but they also sequester some of the alkaloids that the tree of heaven produces and uses that as defense compounds. So tree of heaven is one. They really like red maples. They love black walnuts. Honestly, you'd be hard pressed to find what plants they don't like. They'll feed on plants that kill them, things like milkweed. They just feed on a lot of plants. But grapevines are within that, and we really see high populations of these adult stage in vineyards in the late season, which is a critically important time for grapevine biology. But as I said, all these stages can be present in a vineyard at a given time. And we often are trying to manage all life stages at different points. Now, when you think about what they can do to grapevines and why they're important and why the adults are important, and it's because as we get late into the season, August through October, mid-October, we're trying to ripen fruit. Oh, not we, the grapevines are trying to ripen fruit. And really the fruits become a major sink for resources, for nutrients, carbon, sugars, etc. But not only that, the grapevines are also trying to put away some nutrients for long-term storage. They're perennial plants. They need energy to survive year after year. And this late season period is a really important part for that long-term energy allocation. So as you can imagine, I have a little diagram here. The grapevines are just actively assimilating carbon. That carbon is being taken up. In the late season, the upper canopy is typically the most photosynthetically active. So they're doing a lot of the legwork. Those sugars are being actively assimilated to the ripening fruit, right? We typically harvest grapes between 18 to 25 bricks, and bricks is a concentration of total soluble solids. But sugars it's a proxy for sugars that we use. So we're looking for high sugars. Grapes have a prolific sugar content, and that's really what we want in this late season because it's important for winemaking. But as I mentioned, they're also putting some of that carbon away into those woody tissue reserves. So starch, namely, in the trunks, in the roots, predominantly the roots, but the trunks and the other woody portions make up a big portion of storage as well. So this period is a really critical time for grape health and wine production or grape production. But then the question we have is when we're getting these huge populations of spotted lanternflies moving to the vineyards, and when I say big populations, I mean, 100 plus lanternflies per vine in a pretty large portion of someone's vineyard. And this is what's been documented in Pennsylvania and elsewhere, and what we're starting to see in Virginia too. What we're concerned about is, the lanternflies are extracting a lot of sap, phloem sap. Phloem sap is obviously very high in sugar content, and that's what's being translocated to the fruit. And they're feeding in a way where we're wondering, are they basically competitive sinks for the resources that would otherwise go to the fruit? So sort of like, are they like sucking out what should be going to the fruit or the reserves? So this the concern about the consumption, but also sap-feeding insects largely, like the Hemiptera, are pretty effective at moderating photosynthesis in host plants. So there's multiple angles here. There's the resource competition, but then there's what are they doing on like a hormonal level or a biochemical level to, interrupt or interfere with photosynthesis. And that's what this next slide is. Not only are they taking sugars, but then are they even sort of moderating the actual source itself and the assimilation rate in a way that could have pretty negative implications for grapevines. So these questions informed some past research that I was involved with at Penn State under Dr. Michaela Centenari. She was my PhD, my master is my PhD advisor, and she was organizing and performing a lot of this viticultural research in the early stages of spotted lanternfly, infestations and when it was moving around. And she's still heavily involved, and we work closely together now. And she set up an experiment where we caged individual Riesling vines and exposed them to one season of adult spotted lanternfly infestation. And the goal was, we have growers reporting impacts on fruit ripening, and other things. We need to try to verify or validate what's happening, to the vines. So we infested them with a range of lanternfly densities from about 0 to 200 lanternflies per vine and did this across 2 years. And we infested them during that critical period I was talking about of fruit ripening and late season, storage, which is end of August to mid-October. I'm only going to talk about the gas exchange measurements from this. So if you're interested in the other aspects of the experiments, I'll direct you to our two papers that we've published in PlantArect and Ag Ecosystems and Environment that detail some of these results. But what we did is I took a Cyrus and we measured photosynthesis or single leaf gas exchange on the most mature, recently expanded leaf in the upper canopy. That's a typical practice for viticultural studies. And that's because these leaves tend to be the most active during the late season, and they're also the most sensitive to stress. So if we saw any stress responses, we would think that we would see them there. And then we also use non-invasive sap flow sensors to look at whole vine sap flow and transpiration to try to get this single tissue and then whole vine approach. And what we found was that the function or the impacts of photosynthesis were really dependent on the exposure length of the vines to spotted lanternfly. So these are graphs of just carbon assimilation with carbon assimilation on the y-axis, and then our lanternfly densities on the x. So as you go right, you get increasingly high lanternfly populations. So after 6 days of feeding, we don't see much of an impact. After 20 days of feeding, okay, we start to see a negative reduction in carbon assimilation, and especially in these higher populations. And then as we get to 28 days of feeding, which was the end of our experiment, what we see is that those rates of gas exchange stay low. And not only that, pretty much everything above about 7 to 8 lanternflies per shoot density has sort of having like a similarly inhibited gas exchange. So not only are we seeing reductions, but they seem to be leveling out at some level. And these vines are more or less not fixing much sugar at all. And we did see reductions in sugar accumulation in these vines as well, which is important to note. And then when you look at the foliar transpiration rates, so there's the water use, they tend to be pretty similar to the assimilation data. Not much of an effect in the early part. And then as we get more cumulative exposure, we start to see this reduction in transpiration. Upwards of about 61%, I think, is what we calculated. So a pretty high suppression of gas exchange in this late season. Now, I mentioned, but we also measured transpiration or estimated transpiration using non-invasive sap flow sensors. And the reason we did that is because we wanted to see if the whole vine response matched the single leaf response. So this is sort of a commentary on the important part of, single leaf measurements are great, but whole vine measurements are also really important. And it's good to combine methods to really see what the whole plant responses are, because that might generate some more questions. So we installed these sap flow sensors from Dynamax. And what we found was that the sap flow rates were still affected, but affected at a lower magnitude. So again, after a little bit of exposure, here's the story, not much effect at all or really any effect. Okay, we have a little bit of an effect on whole vine transpiration with more exposure. And then really once we get like 35 to 40 days of cumulative exposure, we see a reduction in transpiration or sap flow. But what's interesting is that the percent relative change from the control, the no lanternfly, to the highest one is much narrower and smaller than what we see with gas exchange. So it's less sensitive, which made us think at the time, well, maybe that means that there's leaves in the rest of the canopy that are potentially compensating or not affected that are, modulating that overall response and transpiration. So that sort of is what's leading into the work I've done here at Virginia Tech. So as I mentioned, heavy adult lanternfly feeding can reduce canopy, I say photosynthesis, but gas exchange more or less upwards of 61%. And then we saw negative impacts to fruit ripeness and that important root starch storage. But what about the rest of the canopy? Grapevines have a lot of canopy. This is probably 1.5 to 2 meters of leaf area. Uh, so or squared of leaf area. So what's going on in the rest of those leaves, and how is the lanternfly affecting those? Because the lanternfly moves around through the canopy as well, and it's not feeding on one spot. They'll feed on one spot, they'll move, they'll feed somewhere else. So it's hard to get a really, you to understand what's happening to the vine, you need to sort of consider the whole plant altogether. So that's one question I have is what's going on in these leaves? And then also, what about other aspects of leaf photosynthetic biology? What's going on with the chlorophyll? Um, are there other things that we can measure that can give us greater insights into what they're doing to gas exchange and just sort of carbon assimilation overall? So to answer this question, or start answering these questions, because there's many questions that remain. Last year, I set up a greenhouse experiment here at a research station in Winchester, Virginia, with a couple different goals. The first one being, what are those old leaves doing in response to spotted lanternfly feeding? And when I say old leaves, I mean not the upper canopy ones that are produced somewhat recently, but the ones that are produced back in June that still happen to be on the vine. We tend to think that they're a little bit slower in terms of photosynthetic activity and maybe contributing less. So what's happening with those? And then also I use this experiment to kind of run wild and just do a range of different measurements, mostly in part to teach myself how to do these so then we can incorporate them into future studies, and also just to really do some fun ecophysiological work with an insect that's fun to work with. So our treatments for this were we collected lanternflies from the wild and then infested vines. You can see I caged a bunch of vines here, and we put the lanternflies in them, and then we just keep up that rate of infestation through the whole period. So 35 days of straight infestation. Any lanternflies that died, I replaced. And then we had a control with no infestation. So we're really looking at like no lanternflies and then worst case scenario. So keep that in mind too, because I'm really sort of stressing the plants out to see what their responses are. And then our field experiments have looked at a range of grapevine varieties. For this, I chose two Vitis hybrids that are important to our industries here in the Mid-Atlantic and the Northeast and the Midwest. So two very popular hybrids called Chamberson and Vidal Blanc. And these have different parentage that include the European grape species or the Eurasian grape species, Vitis vinifera, as well as some native American species parentage too. So a nice way to sort of look at hybrids that have a range of genotypic background. And then using my Cyrus 4, I took a range of measurements. We did a series of just single time point gas exchange survey measurements. I did a couple rounds of chlorophyll fluorescence using dark adaptation. We ran some ACI curves because I really wanted to sort of look at the biochemistry of photosynthesis, and then light curves as well. And that was very limited. I'm not going to present that data today. So really I'm focusing on the chlorophyll fluorescence and the ACI curves today. And then we also destructively sampled the whole vine, and I took canopy leaf area measurements and then submitted those tissues off for macro and micronutrient analysis. And that was to sort of start getting into this nutrition question related to lanternfly too. So I'm not going to present that data today, but that'll be part of a future paper that's tied to this. So keep an eye out for that. So as I mentioned, we did lots of chlorophyll measurements on this. It took me many days to get through all the leaves. We did about 42 leaves per vine, so 40 leaves in total for Chamberson, 36 for Vidal. We chose fully green leaves, so leaves that looked weird, we didn't measure at all. And we just used a single pulse fluorescence protocol that was already pre-established in the Cyrus 4. And what you see is first, the values are quite variable. The Chamberson had a much tighter range, and this is the FV to FM ratio, or sort of that maximum quantum yield of photosystem 2. And the Chamberson range is much tighter than the Vidal. And I think partially what I'm guessing is that maybe at the time, because this was mid-late September to early October, the vines may have actually started to undergo their natural senescence processes, even though I couldn't detect it visually. And maybe some of the leaves I was measuring were already sort of in that early stage, which is why, especially in the Vidal, which maybe is why the range is higher. But interestingly, there's no effect to, or not no effect, and the chamberson, the chlorophyll fluorescence values were higher in the SLF infested vines relative to the control. This surprised me because I had thought that the lanternfly was potentially actually affecting negatively the photosynthesis, like the actual chlorophyll activity themselves, within the leaves. But that didn't seem to be the case, at least for these main leaves. And even though this is statistically significant in terms of an absolute value, we're still looking at above zero point 0.8. So these leaves are functionally healthy still. There was no significant effect with the Badal. And I think that's due to the variability. But this was interesting to me because this means that the leaves are still very much active. And then when you look at the ACI curves, so for those of you who haven't done these type of curves before, what you do is that you take a leaf and you do a series of carbon assimilation measurements at different internal CO2 concentrations. And essentially, the goal is to try to look at the actual photosynthetic biochemical reactions going on and to understand how the leaves perform under various concentrations of saturating CO2. And this was a fun experiment. I actually really enjoyed doing these, and I learned a lot in the process because I hadn't done this type of work before. And first visual take, the curves look very similar. On the left side is the chamberson control. On the right is the SLF. If anything, we see more variability in the lanternfly one. But when you go through and calculate the metrics that you can get out of this, VC max, which is the increased carboxylation rate, and J max, which is the electron transport rate. And I calculated these using a program in SAS from a published paper in Plant, Cell, and Environment. What we see is that there's higher rates for both of these in infested vines. And I'll show you the absolute values in a second. But this also came as a surprise to me too, because I expected photosynthesis to be lower in these leaves as well. And what we're seeing is that it's not, it's higher. And these rates are higher too. So potentially there's some level of compensatory reactions to the lanternfly in these lower leaves to try to maybe balance out the sugars lost by the lanternfly. And we see this with our Vidal Blanc too. The overall assimilation rates are lower in the Vidal Blanc. The chamberson was more photosynthetically active, which we saw with that chlorophyll fluorescence data too. But again, the same trend. A little bit more variability with the lanternfly infested vines, but still this trend emerging that there's increased rates of carboxylation and electron transport. And when you run all the statistics, these emerged as being statistically significant. And these are some of the absolute values right here. So when I compared these to some of the values that have been published for grapes, they're a bit lower than, some of what's already out there. But as a reminder, this data was taken in late September and early October. So we're already approaching that senescence phase. And these vines have, that native parentage too. So I do wonder if their senescence patterns might be a little earlier than some other varieties because they're more tied into the environmental cues. But the point stands though is that the lanternfly vines continuously had higher rates of some of these biochemical parameters. And in the Vidal, we're also respiring more too, which is interesting as well, because then not only are they sort of producing more sugars, or maybe the enzymatic machinery is occurring at a faster rate, but they're also burning more sugars too from the respiration side. So this was some interesting preliminary results and sort of pointed to me that, that those main leaves, so not the lateral shoots at the top, that I measured in the previous experiment in the Riesling field vines, but these main leaves, maybe that they're actually exhibiting greater photosynthetic activity, and this could potentially be a compensation response to try to offset that sugar loss. And I should say too that in the nutrient side, the leaves and petioles of infested vines had greater nitrogen concentration too. I don't know why that is, if they just maintained a greater nitrogen than their controls, and maybe the controls started already remobilizing nitrogen to reserves, or maybe the pressure of the lanternfly sort of redirected nutrients towards that photosynthetic machinery. I think that's less likely. My feeling is that they probably just maintained a higher photosynthetic rate just, to try to compensate. And I mentioned this, maybe they're up-regulating to replace the sugars that are lost to the lanternfly being directly consumed, but also maybe the sugars that are being burned as part of the defense responses to the lanternfly and sort of that modulation of source sink dynamics. defense responses to any pathogen or biotic stress are going to be very energy intensive. And the lanternflies do a lot of damage to the vasculature. And what we've seen in another research is that they're very good at depleting those carbon resources. So not only, is it the removal aspect, but it's also the defense responses that are, but likely reducing the overall carbon balance. So the vines are likely just trying to replace those and maintain some homeostasis. I don't know if that's successful. We haven't measured the starch yet in this experiment, but we're planning to do that this winter. So that's one experiment I wanted to talk about. And then the second is one that we started this fall. That's more sort of an exploration, sort of a seed project of sorts. And where this came from is that we had some really heavy infestations in commercial vineyards in Virginia this year. And I was getting a lot of reports from commercial growers about this phenomenon of this heavy leaf reddening in the upper canopy, where a lot of people's canopies, particularly in the red varieties, because we don't see this in the white fruited varieties because they're not producing as, anthocyanins, um, where people had heavy infestations of lanternflies, their vines were turning red. And so this is a block of Petit Verdot on October 6th. Petit Verdot is a very important variety for us here in Virginia. It's intensely red, the fruit, dark. These vines should be green. They should be the color of these trees back here, right? It's only October 6th, which here in Virginia, fall isn't quite here yet. Well, it is now, but at this time it wasn't. And this is striking because you're looking out onto a block and you see this and you see these symptoms when vines are in severe stress. So everyone's very worried. And then, I was also seeing this in whoops, there we go. In my vineyard too. So this Chamberson on the left is my vineyard. So we kind of thought to ourselves, interesting, let's explore this and let's see if, what's going on here. And the reason we wanted to do this was because typically in vineyards, diagnosing what we call red leaves is really difficult. There's a variety of things that cause this symptomology in vineyards, mostly pathogens. So anything from, important viruses like the grapevine leaf roll associated viruses or leaf roll virus, as we call it colloquially, crown gall, which is a bacteria that can cut off the vasculature and you get this like flame colored scarlet canopy, red blotch virus, and many, many more things can cause this. And this is an example from my own vineyard where, 3 weeks ago, all of a sudden, whoosh, like it turned really red. And likely this is leaf roll virus. This is sort of characteristic leaf roll symptom expression. It's patchy throughout the canopy. You have some leaves that are blotchy that are moving towards this red. And usually in leaf roll infections, you get this striking interveinal green sort of coloration with a dark red leaf. And it's a very economically important virus within the Northeast and really the whole US. And it can come from a variety of sources, but once it's in your vineyard, it can be vectored by mealybugs. So if you have mealybugs, they're going to spread it throughout your vineyard, and that's what I'm experiencing right now. But so when people, were sending me these photos of this red leaves, there was like, is this lanternfly? And it's like, well, it can be a variety of things. So we need to sort of go through and sort it out. So I say this is likely leaf roll because we pulled samples and we're going to test it and to confirm that. But the reason you see this red leaves is that it's likely what we call feedback inhibition. And not only, as I mentioned, you see this in leaf roll virus, but you see this in other pathogen systems like phytoplasmas as well. And what is happening here is that some type of phloem damage is occurring, whether that's due to a virus or phytoplasma replicating, releasing effectors, some kind of phloem damage is happening. The vine may be trying to respond by releasing or activating callose deposition. And then what happens is that the export of sugars slow in the actual leaves, right? And as they slow down and translocation slows, the starch and sugars begin to accumulate in the leaves, and they act as signals that then trigger foliar anthocyanin accumulation. And this is all sort of part of a larger stress response. And I'm summarizing from a lot of other viticultural research just to keep it brief. But then typically once this happens, you start to get a reduction in carbon assimilation. And this is where this is really important, especially with leaf roll virus, is because in many cases, and not always, but sometimes, those reductions in gas exchange then lead to reductions in fruit ripeness and color development. So if you have a grower that has leaf roll virus, in many cases, those vines will sort of hit a plateau of sugar accumulation that might be lower than if they were, asymptomatic. That's not always the case. Sometimes nothing happens, but in many cases, you typically get this lower sugar accumulation. The fruit's a bit pinker. It's not quite what you want. And this is important because we were wondering, is this basically what's happening in response to lanternfly? We know that when they feed on the phloem, they can sometimes damage the vasculature. You can see this visually if you peel back the bark and look at the phloem. There's typically little necrotic lesions. And for a while, we've been wondering, like, maybe there's some level of feedback inhibition going on here, and that's what's causing this red leaf symptoms, which could be important because that might have ramifications for vine health. We don't know necessarily, and it might be dependent, but it's important to explore. So this experiment just took place this fall. And basically, we just wanted to look at what are the rates of photosynthesis and fluorescence in those red leaves versus neighboring green leaves. Are they active? Are they not? What's the concentrations of starch and soluble sugars, as well as chlorophyll and anthocyanins? And what we wanted to look at was really, what's the activity, but also are these leaves just entering a sort of early senescence state, or are they, like where are they in that senescence timeline in relation to feedback inhibition? Because this might have implications for especially our late ripening reds. Many people were seeing that red leaf symptoms when they still had fruit on the vines. And in Virginia, we have multiple varieties that are ripening all the way into mid-October. So if you're starting to see this red leaf symptom expression in early October or late September, that's not good because that might lead to your vines stalling. And I did hear that from some growers where their sugar values just seemed to stop and never progressed. So this might be involved. We're still looking into it. And this was a project that I collaborated with a postdoc scholar here at the AREC, Dr. Amolpreet Kaur Saini. She's a great physiologist and was excited to help me with this. And what we did is we went through and we tagged green leaves and red leaves on symptomatic vines. We only did 6 vines. This was sort of we had to jump on it before they progressed any further. And we were purposely selecting vines that were symptomatic. And then Amol conducted gas exchange measurements 3 times from October 1st to the 18th or 15th. And then one round of chlorophyll fluorescence measurements. And then we also sampled leaves for biochemical assays. And I should say, we're on October 22nd now. So the leaves are changing pretty fast. And now we're really getting sort of fall color expression. So we really tried to track that late season photosynthetic activity and see where it was going and if the leaves were senescing. And what we found was that the leaves that were expressing anthocyanin development, perhaps unsurprisingly, had lower assimilation rates. There was a lot of variability, and that's partially because this is the end of the season and leaf foliar health is quite variable. But both on October 1st and then even on the 15th, the red leaves were consistently lower in assimilation than the green leaves. But you also see, the green leaves are starting to do less essentially too. So we're getting towards the end of the season. So this was an important result. And then also they were transpiring a lot less too. I was pretty amazed at how much the green leaves were still transpiring this late in the season, even though if they weren't assimilating, the normal rates. I think the previous graph I had about 8 micromoles of CO2 per meter squared per second. During peak canopy photosynthesis in July and August, we would expect 18 to 25, very high rates in midday. So they're still, doing something, but the transpiration rates were really high. And I was surprised to see this, but again, as you go to the 15th, things are starting to get lower. It's colder outside now. Conditions are basically getting to the point where they're not really supporting peak photosynthesis. But still, we see this difference between the green and the red leaves. And then the chlorophyll fluorescence data was really variable too. And as you can see, the range of the data was all the way from leaves that were near healthy for the green leaves to leaves that are probably in some senescent state. And then the red leaves were the same way. And this is interesting because visually it's hard to tell if a leaf is senescing right now, or at least when this measurement was conducted, because they looked green. But it looks like maybe the underlying biochemistry is maybe different, and some of these leaves were starting to sort of move towards senescence. But this is why we're doing biochemical measurements too, to see what the different traits were, to try to link those to these measurements. So again, really quick takeaways, and again, this is really, a preliminary data that we're going to use to inform future work, was that the leaf reddening or the leaves exhibiting that reddening were in fact less photosynthetically active. And this tracks with experiments with leaf roll virus, red blotch, and other pathogens that cause this same, symptom. And then the next steps are going to be a whole host of lab-based enzymatic and reagent-based assays, chlorophyll, anthocyanin quantification, and then starch and glucose, fructose, sucrose quantification in leaves. And then really what I'd like to do is maybe order a variety of potted vines of red varieties and infest each one and see the rate of symptom development, because some varieties we have here, to me, express this red leaf symptom a lot faster than some others. So there might be something there to look at. And then also try to link this back, if we can, to fruit ripeness or carbon storage. Because just because we're seeing this doesn't mean there's actual negative impacts on fruit ripeness or cold hardiness or long-term storage. Because as I mentioned, the vine canopies are large. These red leaves are only in certain spots. So, but we want to try to explore that more. So to summarize, can these results shape spotted lanternfly management? The answer is yes and no, right? There's still so much that we don't know about what the lanternfly is doing to the vines, how the vines are responding biochemically, physiologically, gene expression, tissue specific responses. There's so much we don't know. But everything that we do learn and everything that we do study can inch us towards more informed management practices. So even these experiments, you know, trying to understand compensation strategies and linking that to other biochemical or genetic traits, and then comparing varieties to see, well, maybe some varieties are much better at upregulating, carboxylation rates than others. And maybe that leads to an actual tolerance of the lanternfly and its impacts on carbon. Maybe it's important. And I think that can help us move towards identifying if varieties might exhibit some level of tolerance. I don't want to say resistance, because the level of populations that we see in the vineyards are so high that it's going to be difficult to find a variety that's can truly resist 300 to 400 lanternflies feeding on it. But just moving into the direction of maybe being able to identify, hey, this variety is really like vulnerable, so you need to manage it differently. Maybe be more aggressive managing those populations. Whereas for other ones, maybe they can handle a bit more. So trying to get that nuanced information out to growers so they can have a much more informed management strategy. And then also just, improving our knowledge of these late season effects and that feedback inhibition, and then any potential carryover effects that may have, might highlight, the need for different management strategies. If you have a late ripening red like Petit Verdot, maybe you really need to be aggressive managing populations, because if you want those grapes to reach that color and that sugar accumulation, and that you need to let them hang until mid-October, then you might have to manage the populations much more aggressively to try to help them out, help those vines out. But again, it's all a work in progress. There's a ton of people working on this effort. It's not just me. There's a lot of colleagues and collaborators in the Eastern and Midwestern US thinking about this. So, if we're all chipping away and answering all these little questions, we can move the whole industry towards a more sustainable management practice for spotted lanternflies. So that's everything I have for today, and just acknowledgments. These projects are always so much work, and working with insects is difficult and can be variable, and things happen. So a big shout out to all the people at Penn State I worked with, my old advisor Michaela Centenari, and then the whole team of people that were involved then and are still involved in the lanternfly response efforts. They were integral to that research. And then since coming to Virginia Tech, I've had a very welcoming and supportive lab and station staff, and people who are, as excited as I am to work on this project, as well as our funders too, from Pennsylvania and Virginia and the federal government. Without any of that support, we wouldn't be able to do this work. And PP Systems too, for hosting us here, and, providing this equipment. So thank you everyone for coming. I'm more than happy to take questions now or open it up.
Thank you for attending the webinar. To learn more about the CIRUS 4 portable photosynthesis system, visit ppsystems.com.